Morning. All right, let's start. Molecular biology techniques or molecular biology laboratory techniques that is used in clinical labs and so forth for diagnostic purpose. Many of these techniques normally used for research, but sometimes they get used in the diagnostic purposes or even other decision making or determinations or risk factor decision making on the treatment. Okay, so one thing you have to remember when you deciding a technique, what biological sample do I have and what I'm trying to accomplish, okay? And what I noticed, many of these techniques, the principle is exactly the same, but sometimes there's small modification that people, will do in order to fit their need. But the general principle or many of them are exactly the same. So you have to keep that in mind. Especially in the complex test, I've seen some of the questions formulated. But if you don't pay attention very well, you tend to confuse techniques. But one thing you have to remember, the sample that you have and what you're trying to accomplish. Either you have a cell sample, you have a DNA sample, or you have a protein sample or RNA sample. Whatever sample you have to make sure you know what you have first before you move on. That's the key, okay? So as I said, most of them used for diagnostic purpose, determination of risk factors, and the treatment decision to see what kind of changes the person has before you make decision for treatment. Now, the very first one is the centrifugation. You may not call it a technique per se, but in many cases, this is one of the first steps you do in a lab. I mean, centrifugation is something that's commonly done. People take it for granted, but you need it in order to proceed to other things. But in a clinical lab, it has some uses. For instance, if you have a blood sample, you want to separate your red blood cells from the plasma and so forth, or if you want to determine the hematocrit, for instance, you have those little capillaries that you can send the blood to centrifuge it. Now you have your red blood cell pellet, just similar to this one. This is in a tube, but it's similar to this one. Once you centrifuge it, you can measure the pellet of the red blood cells, okay? Which can tell you if the person is anemic or not, okay? Because if you centrifuge and the person hemolytic, sometimes you could see the plasma area is red, which means the red blood cells been lysed and then they're releasing hemoglobin. So it can tell you if the person is anemic or if it's a hemolytic anemic, because you can see the plasma, instead of this yellowish color, will be red if the person is hemolytic. Does that make sense? All right, good. So centrifugation is important. Now, if you look at this person's blood, for instance, you see this clear here. Well, this is a normal blood serum, but if you let or centrifuge, in this case, the person has a white color serum, which represent accumulation of what? Lipids, right? Fats and lipids in the, if the person has eaten and has not got rid of his chylomicron, so you have this accumulation of the lipids transfer mechanism inside the blood, so he has a hyperlipidemia or something similar to that. At least there's a fat in the serum. Is that clear? All right. So that's the centrifugation. Now, the next one is gel electrophoresis. Gel electrophoresis is an important technique, as I always think about what you have as a sample. If you have an RNA sample, or DNA sample, or protein sample, in many cases, the analysis starts with the gel electrophoresis to separate the molecule you have. So in the DNA RNA cases, they are highly negative, right? Why? The phosphate group. So you have a phosphate group, so a bunch of phosphates, so the molecule is already negative. So if you take your DNA sample, you cut them, and you run in the gel, for instance, in this case, they will migrate from the negative end to the positive because the charge on the molecule are highly negative. So then they can go to the positive. Then they'll be separated based on the size, okay? So it's here, it's gonna be separated based on the size. The small size DNA will run faster, and the large ones are hold back, okay? Because they're large, it take them time. They're big, they take their time moving around, looking around. If you wanna recall that they said that if you have a larger molecule, they'll stay back, and the small molecule run very fast around and get out of the jail, okay? All right, so now you separate them, so, you know, nicely, sometimes based on the time to sell. But one thing you have to remember sometimes, and it happened to mostly graduate students who are new, or, you know, a student who's in lab new, if you run your jail and trying to go have a lunch or, you know, come back, and you didn't set your timer to stop so you can, all your sample can run out of the jail. So that can happen sometimes. So keep in mind, so you have to sometimes track it, and then when you start seeing the dye coming all closer to the end, you can just stop, or you can just program it to stop 1 hour. Usually 1 hour, your gel is nicely separated. Now you can cut your gel down for further analysis, okay, that we're gonna talk about. Now, if you have a proteins now that you want to analyze, you see, depending on the sample, okay? Now, your proteins can have all kind of charge on the cell and the surface, right? So now you need to run into the gel. What can you do? What should you do? You want to run your protein. You have a positive charge, you have a negative, you have all kind of charges on them. Or you have no polar molecule that is not charged. So how you make them negative? You denature them with a specific soap called the SDS. So what the SDS will do, it will denature a protein, but also you put a negative charge across your protein. So when you run them, so all your proteins are negative in the same way. So they're gonna run from the negative end to the positive. But now they're gonna be separated based on size, right? The smaller ones run first, and the larger ones are held back, okay? And then you can take those, the gel transfer and analyze further. Okay, if you are interested in specific protein, you can look it up. Okay, so gel electrophoresis, you're gonna see that in many cases when you're looking at RNA, DNA, sometimes in the protein, even though there are other techniques, but for DNA and RNA, in many cases, you have to run the gel and take a look at it. Okay, so when you see gel electrophoresis, when you see running, so that's just the beginning. Okay, but other things can be associated with that, and change the parameter completely. As I said, for further analysis. So is the gel electrophoresis clear? Good. All right. So after the gel electrophoresis, there are a series of blotting methods we're going to talk about. You have what's called the Southern blot, analyzing a DNA sample. Southern blot. Okay? Northern blot, you're analyzing RNA sample, mRNA sample. Okay, so keep that in mind. Western blot, now you're analyzing protein. Okay? Now you have Southwestern blot. What are you doing now? So you're looking at interaction between the DNA and the protein. And what protein bind to the DNA? What's the name of those proteins? The transcription factor. So if you're analyzing a transcription factor, you have that you want to know if you have isolated specific transcription factor, you have a known DNA binding site. So you can take a piece of that DNA and mix them together and see if they run together. So if they bind together, they will run together well. But the key to that one, you run them in a native gel. Because if you denature them, they will not move, okay? All right, good. Now there are east-west, southwest, and all those kind of, but we're not interested in those. These are the four that you should know. All right, so let's move on. So the Southern blot. When I said Southern blot, you're analyzing what? What is your sample you're analyzing? The DNA, exactly. So you take your DNA sample, as I said, it's used for diagnostic purpose. You take your DNA sample, you run it, because they're negative, you don't need to denature them. They run to the gel, Now, once you run to the gel, let's say, for instance, you have your patient sample. You can run sample 1 here, 2 there, and 3. You run to the gel. Now, larger size held back, smaller size up front. After they're nicely separated, then you transfer them to a membrane. Okay? So you take your gel with your DNAs in it, and you have nitrocellulose paper, or then you put it on top of it. Now you run the electricity now on the opposite direction, now from the gel to the membrane. So if that occurs, all your DNA that is separated in your gel will be transferred in the membrane, and they will bind to the membrane. So now they're out of the gel on top of your membrane, in this case, the nylon membrane. Okay? So so the pattern here will be reproduced exactly here. Okay? Now you have your nylon with your DNAs in it. You add what's called a probe. Okay? In many cases, probe is radiolabeled, or probe is basically a specific sequence that you are interested in. Okay? But it is radiolabeled. It is tagged with a P32 radiolabeled phosphate. That will help you to identify when the probe comes and binds to a specific sequence. Then you recognize it, you bind to it, base pairing occur. Now you have, just where your probe's bound. So you expose the membrane to a film, in the dark room, and you develop it. Okay? When you develop it, so now you only have, in this case, 1 band here, 2 band here, which means the probe only recognizes a sequence here and these 2. Okay? It doesn't mean you don't have a DNA here on the others, but this is the portion where your sequence of interest is located at. Does that make sense? So if you want to look at a gene, let's say, for instance, this is a copy of gene. So which means in this patient sample, you have 1 copy of the gene, but in this patient, you have 2 copies, for instance. But in this case, do you have any copy? At least the sequence that you are trying to detect is not present in this sample. It is present, 1 is here, but you have 2 copies here. Is that clear? All right. So in this case, you can look at the mutation, gene amplifications, and many other things you can do. As I said, you can modify these things a little bit to fit your need, but this is the general principle. Is that clear? So that shows southern blotting. Okay, now, and the sickle cell anemia also is detected this way, and many changes have been are detected this way. Okay, come on, move on. All right. Okay. Now the next one is northern blot. Northern blot, what are you measuring, or what your biological sample is? It's composed of what? RNA. So in northern blot, what you're looking at is an RNA basically. And the RNA sample. Let's say, for instance, you have your cell, a specific cell you have treated from this patient, and you want to know if this gene is expressed at any given time. And the gene expression, what is first synthesized? RNA is synthesized, that is later on translated, right? You already seen those transcription, translation, right? When the transcription occurs, you have RNA. When the gene is expressed in the patient or absent, the expression is present or absent, you can measure the presence of the gene expression or the occurrence of the gene expression on the patient's sample, for instance, in the cells, okay? So you take your sample, and then the sample, then you lyse your cell, for instance. You have your cell, but you have to lyse it in order to have the RNA, right? And you can isolate the RNA. Now, once you isolate your RNA, now you run the RNA in the gel, just like I described with the DNA. The RNA is negatively charged, right? So if it's negatively run in the gel, now you transfer to the membrane, just like I indicated. And once you run it to the from the gel to the membrane, now it is laid on top of the membrane, okay? Now, the fact that the RNA, of the genes of interest is somewhere in the membrane. You have to detect it. So what you have to use in order to detect it? Again, you use probe, right? A probe that is radio-labeled, you know. So when the probe is labeled, now the probe is going to recognize the sequence of interest and then bind to it, okay? And once you bind to it, now you take the membrane, you expose that to X-ray film, okay? You develop it. Anybody develop the X-ray? Now take the X-ray film and then go and develop it, and then you see the picture of the person's. Same thing. You put the X-rays in it. The fact that it's radio level, so it has those photons that's released. So they will make a mark on the X-ray film. Once you develop it, then you're gonna see bands like this. Okay. So in this case, in the patient sample, you know, all this, you know, each of these patients have this gene expression at this given time. Does that make sense? All right, good. So that's how you measure. So if the gene is not getting expressed, the genes of interest is not getting expressed, so you will not see any band for that sample. Is that clear? So when you do a northern blot, what sample you analyzing? RNA, right? You separate in the gel first, transfer to a membrane, and you use a probe, okay? So the probe has to be rigid labeled. But sometimes people develop probes that is now radio labeled, but they emit a light. And instead of the radioactivity, just a light. So in case you see those kind of questions, don't be just tied up to the radio label. It can be labeled with a molecule that develops light. And then that also will mark a mark on the film, and you develop it, and you'll see the band. Okay? So when you hear about northern blot, think about an RNA sample. Or if you see RNA sample, think about a northern blot, at least for, as far as I'm concerned. If it's run in a gel, transfer to a membrane, and use the probe, so you have a northern blot. Okay? Is that clear? Good. So in this case, you can look at a gene overexpression. For instance, some transcription factors, such as MYC, as you see MYC here, let me point it out here. All right, as you see MYC here, MYC is an oncogene. It's a transcription factor. It stimulates the expression of genes that are involved in cell growth. So when MYC is overexpressed in your cell, then your cell will continue to grow. And in the cancer case, you know, cancer cells that do not die, but they grow relatively fast, okay? So when the MYC is overexpressed in a patient sample, so you know the cancer may be associated with MYC overexpression, at least to a certain point. Now, if the, for instance, if the tumor suppressor gene is not getting expressed, you guys heard about tumor suppressor gene? Okay. So the tumor suppressor gene, those are gene products that stop cells from proliferating. Now, in a cancer case, sometimes the tumor suppressor gene is not getting expressed. So the person's cells are growing, but you don't have the molecular break, which is the tumor suppressor gene expression. So if it's not present, and you look at the biological sample, your tumor suppressor gene is not present, you can do further analysis, but at least you have an idea that your tumor suppressor gene expression is not present, okay? So at least you look at least the gene expression using this technique. As I mentioned, but you use the RNA sample, transfer to separate in the gel, transfer to a membrane, and this is your label probe. And again, the probe can be rigid labeled, or it could be used labeled by another molecule that just generate light, okay, and then make a mark in the membrane. So here you have all the proteins run in the gel, but only RNA run in the gel, but only recognize your RNA of interest because the sequence match the sequence with the probe. Is this clear? Good. All right. The next one is the Western blot. As I mentioned, biological sample. What you have in this case is protein sample, right? Anytime you hear Western blot, it's a protein sample you're analyzing. But same thing as you see in the DNA and RNA, in the Western blot, Northern blot, and Southern blot, you have to run in the gel. So you take your protein, but one additional step in this one is what? You have to denature your proteins with the SDS so you can have a negative charge on them, right? because the proteins are the native conformation, you have to denature them first, put a negative charge on all across the protein, the peptide, now run in the gel, okay? So they can migrate from the negative to the positive. Now, in our case, what I wanted to show you here, you already ran your gel to the protein in the gel. Now you transfer to the membrane. Now you see your protein sitting in there here. Now you're now using a probe as in the RNA and the DNA sample, the western, northern, and southern. But in this case, your proteins is tagged with a antibody. Okay. Now you use antibody against the proteins of interest. Let me back up. This is your membrane with all your proteins bound to it. But this is our proteins of interest. Now you have to do what's called the staining. Okay? You stain your membrane with the antibody against your proteins of interest. Right? Let's say in our case, if this is, for instance, you take a patient blood and you isolate cells, you put the proteins, and you run it in the gel, okay? In the serum, you run in the gel, for instance, and you have a protein that's specific for HIV, for instance, and you want to identify that. So if the patient has HIV protein, in this case, we have a protein, the P24 antigen. If that protein is here in a membrane, so you add an antibody against that protein, okay? So the antibody, if the protein is present, the antibody will bind to it, right? So once the antibody binds to it, now you have a secondary antibody that recognizes that primary antibody. Okay, that's for more specificity, okay? So that secondary antibody has what's called enzyme attached to it, okay? And this enzyme, when you add a substrate that is uncolored, if you add a substrate, if the enzyme is present, then it will turn it to a color molecule. Okay? And you can see a color development in the band, in the membrane, okay? And then you'll see a band, okay, in the membrane, okay? And that's what you have here. Now, this is, for instance, you have in the line 1, in this case, you run in the gel a known proteins of the HIV virus, for instance, okay? All those proteins here, those are already known, and then you run them next as a control, okay? Now you run your patient sample A, B, C, you run them, next to it, okay? And then you add the antibody, okay? Obviously, the antibody is going to recognize these guys, okay? But look at these proteins, okay? These are from the patient. So the patient C has the P24 here, which is present here, okay? But look at this patient. They also have the P24 present. But how about this patient? Okay, so he doesn't. So now the Western blot is a confirmation test that people use usually after they get ELISA to see the person may have the serum for the anti-HIV or HIV antigen. Now you have to confirm it using the Western blot. Okay, keep that in mind. And we're going to talk about the ELISA, but Western blot is a confirmation test for that. So do you know for sure after you run the HIV proteins and you that is already known, and then you run it next to your patient, and the patient show the same protein expression, so which means the virus is present. Is that clear? Okay, good. Now the membranes usually, it could be nitrocellulose paper, or it can be a PVDF membrane. I don't think that's important for you guys, but in case you heard it, in this case we have a PVDF or nitrocellulose paper for the Western blot. Okay. Now remember, primary antibody and secondary antibody. But some people just use one single. So the primary antibody can also be labeled, but that's not as much specific as when you have the primary and then the secondary recognizing the primary. So we should know, which should tell you that you're really going after the exact protein. Okay. Is this clear? All right. So in the Western blot, what is the sample? Protein, right? Good. And you use the antibody, you run in the gel, and so forth. and transferred to a membrane. All right. This is another example. Now, people, as I said, people change these techniques to fit their need. In this case, there are strips already, that been developed. But in the strip, so basically, this is you run the gel's been run already, and now the proteins known to be from the HIV virus now transfer to a strip, the membrane. Okay. So now you see that disposed nicely here. Okay? So like if people go and buy a pregnancy test, you have a urine sample, and then you have a descent tracker. Similar thing here. Now, in the sample from the patient, you have antibodies against the HIV virus, the specific protein, P24 in this case, or the GAP120. Okay? So you have antibodies that normally recognize those proteins, and those antibodies are in the patient sample, for instance. Okay. Now this step, so all the previous steps that I have said, denature protein, run in a jet, and transfer to the membrane is already done. Now the proteins are already here in the strip. Okay. So now you want to find out if the antibody is present in the patient sample. So if the antibody is present and you add that to the strip, so the antibody from the patient sample will recognize those proteins, the GAP20, 120, and then the P24. So you see here, these antibodies recognize the protein, this protein, and then this protein. Okay? So slightly different from the first one. In the first one, we were looking for the antigens in the sample. But in this case, we're looking at the antibody from the sample. Everybody see the difference? So because if the person is infected, it will develop antibody against the virus. Now you want to identify the antibody in the patient's blood, okay? And that's what and then now it's much more automated. Basically, the first steps are already done for you. Only thing you have to do, take the patient sample and run it and add it to the strip. And the strips already have HIV proteins in it. If the patient blood has an antibody, it will bind to those proteins, and you can develop the color. For instance, here the color will be developed. The color will be developed here. And like in this case, you see the binding occur here, and the P24 also binding occur in these two patients. Is that clear? All right. So, and these are very simple process. People can quickly do that in the lab and get the result right away. But again, you may be required to do one more test, the exact Western blot, in order to make sure that what you're looking at. Not only you see the antibody, but you have to look at the antigen. And the reason for that is because sometimes there are a lot of cross-reactivity. Keep that in mind. The patient may have a sample, some kind of infection from a crazy virus or bacteria from somewhere, and the antibody may look like the HIV antibody. So you may see some cross-reactivity. So now you saw the antibody, you have to look for the antigen to make sure you're getting the right answer. Is that clear? Good. All right, the Western blot. Now, in this case, you are looking at a transcription factor, which is a protein, or you are looking at a mutation in specific gene sequence that the transcription factor binds to. Is that clear? So now, if you take the gene from the person that you are looking at, if the mutation is occurring in the specific gene that, normally the transcription factor binds to it, if there is a mutation, that will prevent the binding of the transcriptional factor, okay? Or if you want to look at if the patient transcription factor is defective or not present, you can mix it with the DNA sequence that it should normally bind and see if the binding occurs. So it's two directions, but the principle exactly the same thing. Let's say in the first case, you want to see in a biological sample, a MYC, for instance, oncogene MYC. Everybody know what I mean, oncogene? Oncogene, the gene expression that causes cancer progression. So if you want to look at a MYC overexpression, so a patient A and a control, patient A, B, and C, you want to look if the MYC is overexpressed in the tumor cell, okay? So if the MYC is overexpressed, you should be able to detect MYC's binding to its specific target if you have the nucleotide sequence for binding site. Is that clear? All right. So you take the sample of patient A, patient B, patient C, the sample containing the tumor sample, and you lyse the cell and you collect the proteins. Now you take that protein sample and you mix with a DNA sequence that is radiolabeled to which the MYC should be bind to. So when you do the mixture, normally the MYC, if the MYC is present, it's going to bind to the DNA sequence. Okay? Now, if that binding occurs, now you take the mixture and you run into the gel. Okay? Now, this is your mixture. You see the transcription factor have recognized this binding sequence and bind to, and you run to the gel. If the MYC is present, the band will shift. Right? If the MYC is not bound, the fact that the probe is smaller compared to the complex, the probe will move a little faster. But the complex will move a little slower because they're larger, so they will move a little slower. So now, if you have overexpression of MYC, what's going to happen? Let's say first, let's back up. If you don't have a MYC expression in that cell at all, what's going to happen? Where are you going to see the band? Huh? Which one? This one or that one? This one? Okay. If you don't have a MYC, it won't be any interaction. So the probe segment will run faster, right? Okay. Now, if you have MYC that is medium expression, so some of the bands will be held back. Now, if you have overexpression of MYC, for instance, what's going to happen? All your probe will be held back. And you can add more probe and see, and you have more binding. So all those probe will be held back. Okay. So which means MYC is there and is overexpressed. Of course, this is not enough. You have more tests you have to do. But this is your southwestern blotting. Is that clear? So you measure, and as I said, you can look at a MYC overexpression, but you can also look at the mutation on the DNA segment that MYC normally should bind on the, or a tumor suppressor gene, for instance, the P53, which is also a transcription factor. If the person has a mutation on his binding site, so you can take an RNA sequence, RNA that normally bind to the P53, express it, basically reverse transcribe it, and to a cDNA and see if the P53 bind to it. If the mutation occurs, so the binding will not occur. Is that clear? Okay. So if there is a mutation on the binding side, the binding will not occur. If the transcription factor is present or not present or defective, the binding will not occur. Is that clear? So this is the way you can detect the DNA and protein interaction. Sometimes there are different names for it. They're called gel shift assay. In this case, that's called southwestern blot. Sometimes people call different names for electrophoretic mobility shift assay, gel shift assay. So all those names. But as far as I'm concerned, so far what I have seen in the medical stuff, they call the southwest blot. Okay. But remember, the name electrophoretic mobility shift assay or gel shift assay. It's the same thing. Is that clear? Good. Okay, now the ELISA. If you recall, it's a quick test. You can detect the presence of antigen or antibody. Okay, so keep it in mind. So ELISA, it's been around for a long time. A very nice technique, very quick to do, and then usually you can do in a large number in a 96-well plate. You see those plates where you have a bunch of, I think I have an example here, like you see here, 96-well plate. So you can, each one of these can contain a patient sample. All right. So as I said, you can detect the antigen from the virus or detect the antibody against the virus in the patient sample. Okay? Are we in that situation here? Keep that in mind because it's two different processes, but they're both similar, but there is a small difference, and that's the difference. Either you want to determine the antigen from the virus, or you want to determine the antibody that the patient has produced against the virus protein. Okay? So the technique is slightly different one way, if you're going one way or the other. So it's specific to antigen determination or antibody against the antigen determination. The antigen from the virus and antibody is from the patient against the virus antigen. Now, it's used for HIV, hepatitis, many other infections, for quick detection, okay? All right. So the first one we're gonna do is the antigen detection. You want to know if the patient blood contains the HIV or hepatitis B antigen, protein from the virus. Now, what you do, you have your 96-well plate. Inside the 96-well plate, you attach an antibody that recognizes your antigen. All right? Here is the key. The capture antibody is inside the well. As you see in the well here, the antibody, this antibody is attached in there. Okay? You synthesize and attach in there. Now, this antibody recognizes the HIV or hepatitis B antigen. Okay? So once you take the patient sample and you contain the antigen, you add it to the well, the antigen will be recognized by the antibody and it will bind to it. All right? So once it binds to it, now you can wash the excess of proteins away from the wells. Now, the next thing you do is to add another antibody that recognizes a different epitope on that antigen. Okay? So when you do that, so you have this sandwich formation, your capture antibody that is already present inside the well, antigen now recognize an antibody that recognizes the antigen. Now you use a secondary antibody that recognizes the primary antibody, and that secondary antibody has enzyme attached to it. And that's where the enzyme link came from. So now you add a substrate to all your wells. So now if your antigen complex is present, the color will be developed, okay? So you take and you measure and you see patient here has 0 color and the patient C has a high color. So that tells you the patient C has some kind of interaction or at least antigen present. Is that clear? Now further testing is needed, for instance, Western blot for confirmation, but at least you know that patient is suspected for either HIV if that's what you're screening for or hepatitis B if that's what you're screening for. Okay, and again, there are possibility of cross-reactivity. Okay, as I said, it could be a protein from another crazy virus or bacteria the patient may carried for a long time or whatever that may be recognized by the capture antibody, and then you have a false positive. Okay. So, but again, as I say, you have to run the Western blot in order to confirm it. That's why when patients positive for ELISA, you have to do a further testing, specifically the Western blot, in order to confirm it. So this was to detect the antigen, the target antigen, right, from the virus. Now the next process now, you want to detect the antibody in the patient sample against the antigen, okay? So what you have, what do you capture in the wells at that point? You engineer what? The antibody or the antigen and capture inside your wells? Antigen. So you take your antigen, you capture, you link it to the bottom of your wells, okay? Your 96 well plate. Now you take the patient sample containing the antibody against that antigen. So you take the patient sample and you add it to your wells. If the presence of an antibody is real, so the antibody against the antigen, that antibody from the patient sample will recognize the antigen and bind to it. Now you have a secondary antibody that recognizes the primary antibody, and you add a substrate. The substrate, when the enzyme is present, the substrate is converted to a product that is generally colored, and you know the patient's blood is at least has some antibodies in it that recognize that antigen. Okay? And again, there are cross-reactivity possible. You have to do the Western blot in order to confirm. All right? So that's your ELISA there. Okay, this is your targeted antibody in the patient blood. All right. So people came out with an even more complex method, and I think I should bring it out to your attention. Now they combine the two together, okay? For you, not only they capture the antibody against the antigen, but But they also have attached some antigen from the virus that will recognize now the antibody. So not only you measuring the antigen presence in the patient blood, you also the antibody, okay? So you measure antigen and antibody together, okay? So now if, in this case, you know, if patient blood contain, not only it contain the antigen, but it also contain the antibody. So now binding occur, the antibody recognize the antigen, in this case P24 antigen, but the fact that it also has an antibody will recognize the antigen that is already captured inside. So basically you are running the two at the same time. Okay, this is a fourth generation. People always come, as I said, modify these things to fit their need. Okay. Now, the sixth method is a karyotyping, which is basically you have, this technique is kind of very interesting. What you're looking at is the chromosomes, okay? If a chromosome has you have a modification or you have a chromosome amplification, if you have multiple copies of the same chromosome and so forth. So you take your cell, for instance, you take your sample from your patients, in this case it's the little fetus, you take an amniotic fluid, the cell from there, which is from the baby, and you grow them. Once you grow them, now they multiply for a few days, and you add a chemical that will stop the cells at the metaphase. At that time, you have all the chromosomes that are nicely formed, okay? So you take those samples, you run it, you centrifuge, and then you take the chromosomes, and you lay down the cells here in the plate and take a look at it. And you can recognize the chromosome, and you look at them closely to look at if you have any abnormality. In this case, for instance, you see this patient here has the chromosome 21. You know, it's supposed to be 2 copies. Now she has 3 copies, which is an indicator of the trisomy in the Down syndrome. Okay. The next one is the Fish Fluorescence Institute hybridization. This technique, to look at a deletion, if the gene is deleted or is amplified and so forth, okay? Now, in this case, again, it's a similar, but in situ, you're looking at inside the cell. So you have a cell on the plate, and you lyse the cell, and then the chromosome is already formed. Now you denature the chromosome. Now you have a probe that comes and binds to the chromosome inside the cell that is basically open, okay? So the probe gets inside the cell or in the tissue. So if you have in situ, it means on site, on the tissue, or in the cell, okay? So you're not denaturing anything, but it's a DNA sitting inside the cell that you are denaturing the DNA from there and add the probes. The probe will recognize the chromosome, this section of chromosome where the probes should bind to. And you have this interaction. So it helps you to detect the chromosome if, for instance, the gene you're looking at, or the gene is amplified, or if it's a chromosome, you have a duplication of a chromosome, or if the piece of the chromosome is break off and moved to elsewhere. All those ways you can look at using the in situ hybridization. So the Down syndrome, you can look at it if you have a 3 chromosome. In this case, you see the 3 chromosome 21 associated that, then you have detected in this patient. Again, it's inside the cell directly or in the tissue. So that's what the name in situ means, okay? And also you can see, for instance, in the chronic myelogenous leukemia, in this case, chromosome translocation, you have changes of piece of the chromosome. For instance, the chromosome 9 accepted piece from chromosome 22 and vice versa. Okay, as you can see, if in this gene, for instance, the BCR is present here and ABL is present here, now you have a rearrangement. Now you have a mixture of the colors. So a piece of this one interact with a piece of this one here and back here. Okay, so you have a BCR ABL complex, and this is not normal. All right, so translocation. So the following one is the silencing the interference RNA. So in this case, naturally we do this degrade the RNAs, okay, from a foreign source, a virus and so forth. Now you send siRNA, which then gonna harness your body natural system to degrade a target protein. Now, the siRNA that you're sending, getting in contact with that proteins, and the proteins, and then interact with the proteins, now it denatured. Now, the sequence of your siRNA will bind to your RNA of interest. And once the interaction occurs, you have a degradation of that, all those RNAs. So basically, it is a way to silence your gene, okay? So that's the gene silencing. And I think you had that siRNA discussion, right? So that's the technique. But you can send a specific RNA sequence that will harness your system now to degrade your target protein, target RNA that recognize that sequence, or that sequence recognized. All right? Now, flow cytometry is another technique that is very, very powerful. You can do so many things, but what you're looking at, it's the cells, okay? But one thing you have to do is the staining. You stain your cells with the antibody, and you run into the machine, and then you can separate the cells depending on the antibody recognizes one cell versus the other. And it is very well used in the immunology to look at the CD4, CD3 cells, especially when a person is suspected to be HIV positive. You can take the blood sample and look at if the CD4 level is low or high, and you can use that using flow cytometry. And the one thing about this machine, you're taking one cell at a time and analyze it depending on the binding of the antibody, the specific antibody for that cell type. And you can analyze them and see what cell types are most present or absent, okay? Or even if there's some other defects, you can see that. As you see here, these cells are very low in the CD8 protein, and these cells also are low in CD3. Now, if you look at these cells are high in CD3, low in CD8, and these cells are high in both proteins, all right? Now, the PCR, I'm sure you heard about PCR before. The PCR, basically, you amplify a DNA segment. You're taking the segment, and then you denature, you add an RNA primers to it, denature your DNA of interest. John, you just do like you do in your DNA synthesis, okay? So the primer will recognize a specific sequence. Now you proceed to a series of reactions. Now, but the reaction complex contains all this stuff: the buffer, the DNA template, the polymerase, Taq polymerase, that recognize a specific, that basically synthesize the nucleotides, and you have your primer. But you also need the DNTP, naturally, because those are the substrate for the DNA synthesis. Okay. In the process, you denature your DNA of interest, you anneal your primer, and your polymerase will then elongate it. But the temperature changes are required for this. In order to denature, you increase your temperature to 95. Now you cool it down for annihilation to RNA to anneal, and then to bind to your primers. Once your primer bind, now you can raise the temperature again for the polymerase to function, and then that can take those nucleotides and incorporate, and your DNA is amplified. As you see here, one cycle, you have, it's several copies that get generated. So this can go on and on. So if you have a sample, or you only have a small piece of the DNA sample, you can amplify and do further analysis, especially with people who's infected, for example, with some virus, but the level is very low. You can amplify and then do the analysis. DNA sequencing, another technique, very important, but you have it, it's a sample, but I want to get to the cloning, because I think that's very, very important. Quickly, if I have time, I can talk about but the cloning, that's one of the techniques. It's not really commonly used in clinic, but it's very, very important. And I think it's developing in drug companies and so forth. For instance, you take a piece of DNA of interest, or a gene, for instance, from your normal human cell, and you can attach that to a plasmid from a bacteria. So once that occurs, you take the plasmid and you transfect that to bacteria, and the bacteria will grow very fast. That bacteria containing your genes of interest, and in this case, the insulin gene, you clone it to a plasmid, transfect it, and then the bacteria start expressing the insulin gene. If that occurs, you have a lot of insulin protein production, and you can lyse the bacteria and collect your insulin protein. But sometimes you can target with a release signal, so not only the bacteria produce the protein, but it can also release in the environment. And this is as simple as that. You can make a component out of that. You take your big tube, you grow your bacteria release insulin outside of the cell, and you can centrifuge them and collect your insulin. And that's what you have. I call that the recombinant insulin. And again, people can modify, but this is a general principle. And you see this modified. Now, instead of using pork insulin, you use human insulin because you take the gene and clone it to plasmid and make that. All right? So this is the gene therapy. You take a piece of gene. You can attach that to virus RNA, a retrovirus, send it to the virus, and transfect that to a human cell. The cell that you have taken from the patient first, you transfect the genes to the virus, and you take that virus, you infect the person's cells with it. In this case, the bone marrow cells. Now, the bone marrow cells that has the genes on it, which can incorporate in their DNA. We call the retrovirus. Once they get in, they incorporate the DNA inside your genome. So now you have your genes of interest that you were lacking first at the first place. Now you have it inside your bone marrow cell, and that bone marrow cell can be injected inside your bone marrow, and so the genes now transfer. So that's a gene therapy. Now the cloning, I'm sure you heard of this. You can take just a regular cells and egg cell from a different sheep. I think you should know this because it's going to be, I don't know, human cloning is far away, but at least it's been done in in sheep. So you take this cell, any cell, in this case a harder cells, that is diploid, and you take an egg from another animals. And the egg cell, you take the DNAs out of the egg. Now, from this sheep, you take the DNAs out. So this empty shell now with all the proteins and the DNA, you fuse that with the cell from the udder, which is already a diploid, okay? Now you have fusion. Now the cell will reprogram to become an embryo, okay? And you take that embryo, now you place that back to the sheep here, the surrogate sheep. Now this embryo has no genes belonging to this sheep or this sheep. Everything has come from this sheep, and then you have a new animals here that is completely different from these two parents, but naturally from this. Okay, and then they have sheep that just synthesize the clotting factors, the human clotting factors, and so forth. Okay, and that would be possibly the next step. I don't think it's going to happen because it's really unlikely to occur. It's a human cloning. But anyway, I just think you should know the cloning techniques. If you don't have any question, thank you. All right. For the sequencing, look at it. It's not that crucial, but really I think it's very simple. But I'm not going to hold you responsible.